In this talk, we will learn about the transport of sugars in vascular plants. The growth of all organisms require water and nutrients. The acquisition of nutrients and water is easy in unicellular organisms or in cells in general. This acquisition can happen through passive transport, where we can have solutes or substances move from a high concentration gradient to a low concentration gradient. In the context of a cell, this can happen directly through the plasma membrane through diffusion, or we can have proteins present in the plasma membrane that facilitate this movement and hence is called as facilitated diffusion. Passive transport does not require any energy, and the flow of transport is always from the region of high concentration to the region of low concentration. Active transport is another transport mechanism which requires energy. In active transport, solutes or substances always move from a region of low concentration to high concentration. The acquisition of nutrients and water is, however, more challenging in multicellular organisms. Plants are examples of multicellular organisms who are photoautotrophs. They are able to acquire water and nutrients by different mechanisms. This includes passive transport and active transport that is normally used by the cells of the plant. We also observe the phenomenon of bulk flow, which is observed in the entire organism. Bulk flow is a type of long-distance transport where the transport of fluid is driven by a pressure gradient. Thus, the fluid and the substances dissolved in the fluid are able to move from a region of high pressure to a region of low pressure. Plants do not possess a pump, which is observed in some animals. In some animals, we are able to see the transport of fluids using a heart which functions as a pump. This is absent in the case of plants. Plants have different routes to transport substances from one cell to another cell. The apoplastic route is where the substances are transported through the cell walls and extracellular spaces of the plant cells. In fact, apoplast means everything that's external to the plasma membrane. Thus, it includes the cell wall and extracellular space. However, in case the cell is dead, the apoplast also includes the interior of dead cells. The symplastic route, on the other hand, involves the transport of substances through the cytosol of the plant cells. The water and solutes cross the plasma membrane of a cell only once to enter from the environment, and once they're inside a cell, these solutes and water can travel through the plasmodesmata of the plant cells. The plasmodesmata are pore-like structures that are present between neighboring cells. They allow for the transport of substances between two cells without involving any transport outside the cell. The symplast is the cytosol of all the living cells, which includes the plasmodesmata. The transmembrane route is where substances repeatedly cross the plasma membrane as they pass from one cell to another cell. Sugars or carbohydrates are important molecules that have diverse functions in a cell. They can be oxidized to generate energy molecules like ATP and NADH. They can also be used as building blocks for other macromolecules, including nucleic acids. Different organisms have also used sugars for protection purposes. For example, the cell wall of plants is made up of cellulose, and cellulose is a sugar or carbohydrate. There are different organisms that are able to make carbohydrates like starch and glycogen that help with storage of glucose, which can then be used later to obtain energy. Sugars are essential for all living organisms. The intake of sugar is easy in unicellular organisms because they make proteins present on the cell surface that promote the uptake of sugar from the environment. It is, however, more challenging for multicellular organisms, and hence they must evolve special structures or systems to obtain and transport sugar. In the case of plants, plants have evolved a shoot system. This is especially seen in vascular plants. The shoot system produces sugars by photosynthesis and also plays important roles in supporting the plant. Photosynthesis is the process of making organic carbon compounds from inorganic carbon dioxide. The energy to drive this process is obtained from light. Photosynthesis can be performed by many different organisms, including prokaryotes. Cyanobacteria were the earliest organisms that had the ability to perform photosynthesis. Among eukaryotes, we do have unicellular protists that have the ability to perform photosynthesis. Among multicellular organisms, algae and plants are known to perform photosynthesis. All organisms that are able to perform photosynthesis are primary producers. They're able to make the organic carbon compounds that support the life of heterotrophs. Vascular plants have evolved organs, and one such organ is the leaf. Photosynthesis occurs in the leaves of vascular plants. Within the leaves, there are mesophyll cells. These mesophyll cells contain chloroplasts, which are membrane-bound organelles. Within the chloroplast, there are disc-shaped structures called as thylakoids. These thylakoids have pigments that enable the process of photosynthesis. The thylakoids are stacked one on top of another, and each stack of thylakoids is called as a granum. Multiple stacks of thylakoids are called as grana. The thylakoids are suspended in a cytoplasm-like matrix that is present in the chloroplast, which is called as the stroma. Photosynthesis can be represented by a chemical reaction which shows that carbon dioxide and water, along with light, are able to produce sugars like glucose and oxygen. This is the reaction for what we call as oxygenic photosynthesis. Photosynthesis is a multi-step process. There are two main sets of reactions that occur. The light reactions occur in the thylakoids of the chloroplast. The second reaction is the Calvin cycle, which occurs in the stroma of the chloroplast. In the case of plants, photosynthesis is a self-sufficient process. The light reactions produce the energy that is needed to drive the process. The energy in photosynthesis is obtained in the form of light that is used to make ATP and NADPH. The light-dependent reactions of photosynthesis are driven by photosynthetic pigments and light obtained from the sun. The photosynthetic pigments are able to absorb light present in the visible range of the electromagnetic spectrum and release electrons in response. The most common photosynthetic pigment observed in plants is chlorophyll. Plants in their leaves have light harvesting systems that are able to obtain energy from sunlight and transfer that energy to the photosynthetic pigments. These light harvesting systems are called as photosystems. Photosystems are a collection of proteins and photosynthetic pigments. They have two main components. The light harvesting complex is the part of the photosystem that helps transfer energy obtained from sunlight to the second component, which is called as the reaction center complex. The reaction center complex has a pair of chlorophyll molecules. These chlorophyll molecules obtain energy from the light harvesting complex and lose a pair of electrons. There are two photosystems that are present in chloroplasts, and both systems are in the membranes of the thylakoids. The light reactions begin with photosystem 2, which is present in the membrane of the thylakoid. Photosystem 2 obtains energy from photons present in light. It absorbs the energy and loses a pair of electrons from the chlorophyll present in the reaction center. The electron pair is then transferred to plastoquinone, and then to the cytochrome complex. This transfer of electrons results in the transport of H+ ions from the stroma of the chloroplast into the thylakoid space. This causes an accumulation of H+ ions, resulting in a high concentration of H+ ions inside the thylakoid and a low concentration of H+ ions in the stroma of the chloroplast. The H+ ions that have been transported into the thylakoid space can be passively transported outside of the thylakoid using the enzyme ATP synthase. This passive transport of H+ ions releases energy, and this energy can be used by ATP synthase to make ATP, which is an energy-containing molecule. The ATP is synthesized in the stroma of the chloroplast. The electrons that were lost by chlorophyll have to be replaced in photosystem II. Additional electrons are obtained from water, and the water is split in such a way that the electrons are given back to chlorophyll, and in the process, oxygen is produced. Similar to photosystem II, photosystem I can also use photons present in light that results in the release of an electron pair from the chlorophyll present in its own reaction center. This electron pair is then transferred to ferredoxin and ultimately to NADP reductase. NADP reductase then transfers the electron pair to NADP+ to form NADPH. The NADPH that is made from photosystem I and ATP, which is made from photosystem II, are used to drive the next set of reactions which occur in the Calvin cycle. Both of these are molecules that are able to store energy that can be used later. The electrons that are lost from the chlorophyll in photosystem I can be replaced from the electron pair that was released from the chlorophyll of photosystem II. As we observed previously, the electron pair from photosystem II is ultimately transferred to the cytochrome complex. From the cytochrome complex, it can be transferred to plastocyanin and then to the chlorophyll that is present in photosystem I. The next set of reactions in photosynthesis occurs in the stroma of the chloroplast. The Calvin cycle is a series of reactions where carbon dioxide from the air is fixed into organic compounds. The carbon dioxide in the air is added to ribulose 1,6-bisphosphate by the enzyme called as Rubisco. From there on, a series of reactions occur which ultimately result in the formation of glyceraldehyde 3-phosphate. The glyceraldehyde 3-phosphate is then used to make other sugars like glucose, fructose, sucrose, etc. To drive these reactions, energy is needed. This energy comes in the form of ATP and NADPH that were synthesized in the light reactions of photosynthesis. All the reactions related to photosynthesis occurs primarily in the leaves of plants. They actually happen in the mesophyll cells. In the case of angiosperms, we have two types of mesophyll cells, and photosynthesis primarily happens in the palisade mesophyll cells. Once photosynthesis occurs in the mesophyll cells, the sugars are made. Thus, the leaves of the plant, which has these mesophyll cells, is the source of sugars and hence is rich in sugars. The parts of the plant that consume or store sugars is called as the sugar sink. This includes flowers, young fruits, or even roots of the plant. The carbohydrates that are produced by photosynthesis will enter the phloem, which is present in the veins of leaves. Phloem is a type of a vascular tissue. Phloem is the vascular tissue that transports food like sugars throughout the plant. The cells of phloem are alive, unlike the cells of xylem, which are dead at maturity. When we look at the different types of cells in phloem, There are sieve cells that are normally observed in gymnosperms and seedless vascular plants like ferns, while angiosperms show the presence of sieve tube elements that are sometimes also called as sieve tube members. The sieve tube elements do not have any nucleus but are associated with companion cells that do have nuclei. These companion cells have plasmodesmata that allows them to communicate and exchange material between the sieve tube elements. Thus, even though the sieve tube elements do not have a nucleus, they are able to take ribosomes, proteins, and other material from the companion cells. At the end of the sieve tube elements, the wall has many pores. This region is called as the sieve plate or sieve area. In order to reach the phloem, the sugars have to move through different types of cells. This includes the mesophyll cells, where they were made and hence are the source of the sugar, the bundle sheath cells, which is a layer of cells surrounding the vascular bundle in the veins of the leaf, and other types of cells until they finally reach the sieve tube elements of phloem. The sugars are transported by the symplastic route, or in some cases, a combination of the symplastic and apoplastic routes. Companion cells that are in close proximity of the sieve tube elements can enhance the movement of the sugars between the apoplast and the symplast. In some cases, the sugars have to be transported into the phloem cells through an active transport process. This usually occurs in two steps. There is a proton pump present on the plasma membrane that is able to transport H+ ions out of the phloem cells. This creates a high concentration of H+ ions outside the phloem cell and a low concentration of H+ ion inside the phloem cell. A cotransporter then allows the passive transport of H+ ions back into the phloem cell. This passive transport releases energy that is able to drive the transport of sugars like sucrose from the outside of the phloem cell into the phloem cell. This results in a sugar-rich fluid that's present in the phloem, which is also called as the phloem sap. The sugars in the phloem sap then are able to move to the remaining part of the plant through a process called as translocation. Phloem sap is able to move through the sieve tube elements by the process of bulk flow. In the case of bulk flow, the movement occurs from a region of high pressure to a region of low pressure. In the case of a plant, the high pressure region is the source where the sugars are made. Examples of sources are called based on the pressure flow hypothesis. The production of sugars at the source results in a high solute concentration in the phloem that is near the source. This high concentration causes water to move from the xylem to the phloem through the process of osmosis. This movement of water increases the pressure in the phloem that is near the source. This increase in pressure pushes the phloem sap either upwards or downwards in the phloem. At the sink, which are regions that are low in sugars, the sugar that is present in the neighboring phloem will leave to enter the sink. This causes a drop in the solute concentration in the phloem that is near the sink, which results in water leaving the phloem and entering the xylem or other neighboring tissues. This water loss creates a low pressure area near the sink. Thus, the changes in pressure in the source and the sink drives the bulk flow of phloem sap. As the plant grows, there can be situations where there are more sinks than what the sources can support. In such situations, the plants go through a self-thinning process. In this process, the plants remove sinks like flowers, young fruits, etc., which allows the plant to sustainably share the nutrients that it is synthesizing through photosynthesis. The removal of sinks is also used in horticulture, where farmers remove some fruits so that the other fruits can grow larger. It should be noted that phloem is close to the periphery of the stem in a plant. It is the outer part of the vascular tissue, and as we can see in the figure, it is near the stem during primary growth, and in secondary growth, the primary phloem is closest to the periphery of the stem, while the secondary phloem is underneath the primary phloem. Thus, any injury to the outer epidermis or the periderm of the stem will cause the phloem sap to ooze out of the plant. Insects, in fact, take advantage of this location of the phloem and bore into stems to obtain the nutrient-rich phloem sap. With this, we come to the end of our talk, where we learned about the different transport routes present in plants, as well as how photosynthesis occurs in plants. We then learned about phloem and the concept of the source and the sink. We learned about the loading of sugars into phloem and the pressure flow hypothesis that explains how sugars are transported in phloem.